if I need to post that. Oh, all right. Here we go. So we went through all of this stuff yesterday, looking at all of these basic things, right? The bacteria. We got through further than the other classes, but we still want to review like we always do. Eliza, we talked about this archaea bacteria. It was the first kind of bacteria that we talked about. And what is another name for archaea bacteria? What is another name that we gave for that? The type
extreme thermo something. Thermo. I don't know.
Okay, well, extremophiles. And
yeah.
what I want you, yes, what I want you guys to do is if you have your notes open or whatever, you know, I want you to, just like in class, I want you to exercise your minds and see what you can remember. If you can't remember it, that's fine. I'll just keep going down the alphabetical list. Okay, so you said extreme thermophiles. What does that mean? Tell me about those. That's Eliza. I still want you to talk about those. What does that mean that those bacteria can live where? Where would we find them?
Like really hot places like black smokers and environments like that, and that's where they thrive in the most, unlike other cells.
Very good. Like in these aquatic environments, very hot, like hot springs and black smokers. These, what should happen? What should happen to a normal cell, Nicole, if it's put in such high temperatures? What should happen to a normal cell if it's put in such high temperatures? Other than the fact it should die or be cooked or whatever.
I was gonna say denatures.
Okay, it denatures. What are the macromolecules that we learned about that denature?
Why is it doing that?
Do you have headphones on? If you have headphones in, it won't echo. Your microphone is picking up your speaker. Okay. So it's proteins. So it's the proteins, essential proteins in the cell that would denature. Obviously, if it's at such high temperatures, the whole cell is going to just fall apart. But I want you to make the connection between what we learned in the past and what we're learning now. Something that we talked about at the very beginning, whenever I said that I'm going to go ahead. Let's use magenta. And the microorganisms down here are something called chemoautotrophs. So if you want to scribble that down somewhere, okay? So these are found at the black smokers. I didn't write this down yesterday, did I? They're found at the black smokers, and they use sulfur to produce energy. Oh, energy. You get it, photosynthesis or okay. So this is what's special about these extreme thermophiles is that the bacteria that live down here are what's called chemoautotrophs, and they're going to be found at the black smokers. And instead of using cell respiration or photosynthesis, they actually process sulfur to be their energy producing. That's what they use to produce their energy. Okay? You got it? One of these things to scribble down if I haven't given it to you already. I know first period doesn't have it. I wasn't sure if I gave it to you guys or not. Okay. Caitlin, what kind of extremophiles are these? These are extreme. What's the environment that they live in if you can't give me the actual word?
They live in really salty ones, but I don't remember what the environment is called.
Okay. They live, so it's very, very salty. Does anybody remember what these are called? It starts with an H.
Halophiles.
Halophiles. Awesome. Extreme halophiles. And it's just high concentrations of salt. So very high salt. Right? We have this from yesterday, right? I'm just writing it down again. So what should happen to a regular cell? What should happen to a regular cell? Caitlin, can you share that? Do you remember about the environment?
It'll go through osmosis and lose all of its water and like shrivel up and die.
Perfect. Give me the H words. Inside the cell versus out.
The inside of the cell is hypotonic and the outside is hypertonic.
Awesome. Okay. So on the test, whenever I say what should happen to a normal cell in a extremely salty environment, it should shrivel and die due to osmosis because the interior of the cell is hypotonic and the outside is extremely hypertonic. Okay, making those connections. See, tests in the past, you're not allowed to fail it and forget it, or you're just not allowed to forget it. Okay, found everywhere. The shapes. Can you tell me, Carly, the 3 shapes of bacteria, just generally?
Isn't a rod, a spiral, and like a circle?
Okay, a circle. So of those 3, can you tell me the actual bacterial name for those shapes? Any one of them?
I'm gonna try not to look at my notes, but spirali?
Okay, spirali.
Yes.
Bacilli, is that going to be the round or the rod? Bacilli.
Rod.
It's going to be the rod. And then the last one, which is the round, that's the cockle bacteria, right?
Yes.
So that's what we're looking at for these things. Just refreshing, because there's not really a clear way I can ask these, but make sure you know, right? Monococcus are just simply cockle, that's 1. Diplo, 2. Staph, a clump. Strep, a strip for the cockle bacteria. Bacillus, single. Diplo, strepto. Make sure that on the test, when I say you are infected with a staph infection, you know that it has to be a cockle bacteria because there is no staphylo anything else. Okay? All cells have to be small. Okay? All cells have to be small. Why? Why is it important? Well, Madison, why is it important? Every cell gets its food. How? How does every cell get its food or its gases? Where is nutrient transport or gas exchange? Where does that take place in every cell? Madison, I don't know if you heard your name.
I don't know.
Okay.
I can't tell you without looking at my notes.
Okay. Well, it's right here. It's saying about the membrane. You have to have, so every cell gets its food or nutrients, or it has to pass through the membrane. All right. So it's important, right? We talked yesterday as far as surface area to volume. If we look here, large volume, that's going to be a smaller surface area, right? Than if you have the same volume and a bunch of other smaller cells, you have a larger surface area. So generally, food is distributed through the cell by Owen.
Diffusion. Oh.
Okay, Madison, you got it, Madison? Was that you? Diffusion. Okay. So Owen, if there's too much volume, the outer membrane can't what?
I don't know.
You don't remember?
No.
So if it's being absorbed through the outer surface, and if we have a large volume, okay, and it can only distribute through diffusion, I don't have a big enough membrane, the outer membrane can't do what? Carter?
Supply enough nutrients.
That's it. It can't supply enough nutrients. It's not big enough to supply enough nutrients because the plasma membrane, the surface area, isn't big enough to supply the big volume inside the cell. Okay. So, Lane, why? So then we did say, like, all cells need to be small, but eukaryotic cells are able to be about 100 times larger than prokaryotic cells. What do they have that prokaryotic cells do not?
Membrane-bound organelles.
Membrane-bound organelles. They have membrane-bound organelles. Because they have those membrane-bound organelles, right, they're able to create this endomembrane system, okay? So we already talked about all cells must be small because a couple slides ago, they have that membrane-bound organelles. It creates the endomembrane system. And then, Emma, what is that then causing to happen? If we have an endomembrane system, this increases what? Within a
The
cell.
surface area of the
Yeah.
membrane.
It increases the overall surface area of the membrane. Okay. It increases the overall surface area of the membrane. So that's what something to add. If we were to go back, looking at this teeny tiny, right? This is that these don't have membrane bound organelles. So that's why this is how small a cell would have to be in order for the nutrients to diffuse thoroughly throughout the cell so that cell can get the nutrients available. So membrane bound organelles allow for eukaryotic cells to be so much larger because it essentially increases the surface area of the cell. Okay. And oh goodness, we did go through all this yesterday. Okay. 2 types of cells, gram positive, gram negative, and it's based on their cell wall. Okay. I'm just going to review this with you guys instead of asking you questions because there's so much. You, again, can go back to this slide and write down all these notes. I definitely, when I'm saying as far as the cell wall and I have peptidoglycan, I do want you to know that. You are going to need to know peptidoglycan because it's going to be really difficult to explain the differences between these if you don't know that one. Okay, you have to know that the gram positive is has that thick peptidoglycan layer. If you don't already have your notes out again, or go ahead and grab those out of there so you can add any information or review this as we go through it. Okay, gram negative. So the gram positive, those ones are kind of easy to kill with antibiotics. And the gram negative, not so easy because it has a more complex cell wall. Okay. All right. So yesterday, when we talked about these, we talked about the process of the staining. Whenever you do the gram staining, you would scrape your teeth and you would, it's so cool. Maybe we'll do it anyway. We'll go break the rules. Smear that on your slide. You would heat fix it. So you'd put it over a flame real quick. And then you would add grams iodine to it. You rinse that off. And that's the beginning step of the staining. And then you would add this crystal violet, which is the purple. Then you would rinse that off. And then you add this stuff called safranin, which is the pink. All right. Well, the purple, the crystal violet, reacts with the peptidoglycan. Okay. So it reacts with it. So it stains the peptidoglycan. Well, why is the gram negative not stained purple? Alina, why do you think the gram negative is not staining purple? if it interacts with the peptidoglycan? Why do you think the gram-negative doesn't stain?
I'm not sure.
Okay. Liza, do you have a guess?
Maybe because it's like surrounded by more membrane, so it's harder to get to it.
Yeah, it can't get to it. It's blocked. So what happens here instead, so you go through the series of stains, the purple stains the peptidoglycan and the pink stays within that outer layer. Okay, so it's the other protective layer outside the peptidoglycan layer. Okay, so we could just call it an outer membrane outside. Okay. So I gave you guys extra notes yesterday as far as talking about how specific antibiotics work. So looking at your notes, Kate, I'm sorry, Nicole, do you have anything as far as how penicillin acts on gram-positive bacteria, how it's able to help kill it? If we have, you can say no, but you should have this in your notes at some point. And we're looking at what do the basic. Okay. Nicole, turn your mic off. Okay. I'm gonna move to, because I don't know if you heard it, but you were giving us a very shrill feedback. So I'm gonna move to Caitlin. Caitlin, how do the basic antibiotics such as penicillin, what do they do to kill the gram-positive bacteria?
I don't have anything about that in my notes.
Did we not write that yesterday? Anybody?
The last thing I've written is it talks about antibiotic resistance or well on their way or becoming resistant.
Yeah, that's after that.
Oh, it's after that?
Yeah. Oh yeah.
Let me look.
Yeah. It's whenever we were talking about the gram staining, there should be, because that's what I wanted you guys to write down. If not, obviously I'll go over. Does anybody have it?
No.
No. Well, okay. So what you do need to know is, and it's very similar to the way the stains work, all right, is it has to do with that peptidoglycan layer. So the antibiotics such as penicillin or streptomycin, they break down the peptidoglycan layer. Okay, and that allows your immune cells to get to the plasma membrane, actually attack the bacteria. So just like the gram stain, that crystal violet can't get to the peptidoglycan layer, same thing with those antibiotics. They can't get through that protective outer layer. So they can't get to break that down. Okay? I'm not sure if it's spelled right. All right. So just very basic, right? The penicillin breaks down peptidoglycan layer. It can't affect the gram negative because it can't get to that peptidoglycan layer to break it down. Good? Going? Okay. So we talked about glycocalyx. And the gram-negative cells have this. And that's the protective layer that's outside the cell wall. And there are glyco, sugar, calyx coat, sugar coat. And there are two chemical compositions, capsule and slime layer. Knowing that the capsule is firm, right? Just like a capsule that you would take for medicine. And then a slime layer is loosely attached, which makes sense because it's slime. Okay, pictures of each, taking a look at those. And then talking about this Della Vibrio. So this is, like you said, this is the last slide that you have any notes on. Actually, I think we got all the way to motion. I think that's where we ended, was looking at those. Maybe not. Okay. Since gram-negative cells are so difficult to attack with antibiotics, looking at this other microorganism, that what it does is it can break through that glycocalyx. It actually attaches to it and kind of burrows through it, and then eats the cell. And these are the things, and it says that they are either already antibiotic resistant or well on their way of becoming resistant. So science always is looking for ways to fix the problems that we have. So even though it keeps saying things are, oh, it's antibiotic resistance, we'll know that there's always something on the forefront as far as research is making sure that we don't die because of antibiotic resistance. Okay? All right. So we talked about, I could have sworn we got this far. I should have written it down. Two external structures. So like I said, there's not every bacteria have these. The fimbriae specifically, they're simply to attach to a substance, and it's kind of like Velcro, but not every bacteria have something like that. And this is something called a pilus that we'll talk about later when we talk about genetic recombination. And this is one thing called conjugation that we're going to get to. So just simply wanted you to know what these are. So when you have a diagram, you'll be like, oh, this is the fimbriae and this is the pilus. Okay. As far as them moving, right? They can move through flagellum. They can just corkscrew, and they can kind of squirt themselves along with like jet streams of slime. And then this one, chemotaxis. Okay, chemical taxis, to move in response to a chemical stimulation. So whenever you're thinking as far as how else are bacteria or other microorganisms, how else are they going to communicate? Because they have to communicate. So just think about yourself, that you carry out chemotaxis as far as scent. If something's in the kitchen and it smells amazing, that chemicals are reaching your nose. You're probably going to make your way to the kitchen and be like, when are the chocolate chip cookies done? Or if someone's bacteria in their gut is very unhappy and producing a noxious odor, that you're going to chemotaxis out of the room, right? You're moving away from that. So that's what that is. It's simply when bacteria are moving towards or away from a chemical stimulus. So examples of these: moving with a flagellum. Did I show you this yesterday? Somebody?
I don't think so.
Okay, because it just really seems like I'm doing a rerun here, but I did. No, I showed it to the other kids. But this is, I said, there's so many different. Our microscopes are so basic. This is a fluorescence. This is using, this is a fluorescent microscope. So what this is showing is something we would not be able to see, but this is just showing the movement of the bacteria, how random it is, and a little rainbow trail that it would leave behind, in the motion that as it's moving with its flagellum. So just thought that was cool to share that with you guys. Then coccobacteria, they're just spinning. They're crazy. Look at these. Maybe. There we go. Come on. That's crazy. And then the Del Vibrio is smelling out the bacteria. And the question is, so whenever we're looking at this, it's saying that it's blah, blah, blah. This is a good read anyway. But what we're looking at is how trying to figure out the Del Vibrio, how it's finding out the prey, how it's looking for it. So this is it just randomly looking for its prey. So obviously they're trying to figure out how to make that better. Okay, so anyway. General structures of all bacterial cells, going back to that, structures that are common between prokaryotic and eukaryotic cells. Carly, what are some structures? Have I called on, like it's your turn, right?
Yes.
Okay. What are some structures that prokaryotic and eukaryotic cells have in common?
They both have a membrane.
Yes. Okay.
And don't they have a cell wall? And then that's similar to plants?
Not all. Because plant cells have a cell wall, but animal cells don't.
Yes. Okay.
What else?
Ribosomes.
Mm-hmm.
Cytoplasm.
Mm-hmm. And then one that people don't normally think of.
I want to say DNA, but
Yeah, good job. I was going to say genetic material, but go ahead. That's it. DNA. Yay. Okay. All right. Time to review. We did this already today. So just whenever you go back to, as far as study guide, you know that these are the things that we've covered up to this point. Number 11, I need to add this is horribly written as far as, okay. So the common structures between eukaryotic and prokaryotic cells. So everything before this you need to know. Okay. Here's where it gets fun. So when looking at the genetic information in bacteria, it is pretty simple. It's basic. So again, this is a slide that you do not need to write down all the notes right now. You do know that prokaryotic organisms do not have a true nucleus. Only eukaryotic organisms do because it's a membrane-bound organelle. They have what's called a nucleoid region, and that's simply the concentrated mass of DNA, 1,000 times less than what's found in eukaryotic cells. In this slide, you see it's like a ring. Well, obviously, I want you to picture that like it's a rubber band. And it's not just hanging there in the bacterial cell. It's like a rubber band. And if you kind of crumple it all up, it's just going to be in this mass. And that's what's called the nucleoid region, where that mass is found. Okay. Single circular non-homologous chromosome. Non-homologous compared to chromosomes of eukaryotic organisms. They're linear and occur in pairs. This should look familiar. Okay, so the bacterial DNA is a circular, non-homologous. And this, you should remember from the bio, this is called a karyotype, and it's just lining up the homologous chromosomes of a human, right? So it's saying they occur in pairs, right? One from mom, one from dad. And they are linear, right? They're not one big giant circle. There are a bunch of pairs, and that is pairs of linear homologous chromosomes versus bacteria, circular, non-homologous. As far as other genetic information, in bacteria, they have these little things called plasmids, which we've really taken advantage of. Plasmids have extra accessory genes. And that's what science has taken advantage of to make genetically modified organisms. If you're doing microbiology later, you'll have a lot of fun working with bacteria and messing with those plasmids. Okay. Bacterial cells undergo asexual reproduction, which is called binary fission. All right. So asexual reproduction, binary fission. Okay, and I want you to think it's just like mitosis. So if you remember mitosis at all from bio, you have a, oh, are we done? Is class over?
Yeah.
Okay. Okay. All right. I need to have a clock just like right here so I can see. So, all right. So what we're going to start with then on Monday is we'll talk about binary fission and we'll keep going from there. All right.